This paper's on spectral quality of light modulates emotional brain responses in humans. It's also by Giles van der Waal. He's got a team as well working on this one. It's the second most recent he's published. Cyclotron Research Center, University of Liège, B4000 Liège, Belgium, Geneva Center of Neuroscience, and Swiss Center for Affective Sciences, University of Geneva. It's just listing different institutes that participated and contributed. Light therapy can be an effective treatment for mood disorders, suggesting that light is able to affect mood state in the long term. As a first step to understand this effect, we hypothesized that light might also acutely influence emotion and tested whether short exposures to light modulate emotional brain responses during functional magnetic resonance imaging, so an MRI. 17 healthy volunteers listened to emotional and neutral vocal stimuli while being exposed to alternating 40-second periods of blue or green ambient light. Blue relative to green light increased responses to emotional stimuli in the voice area of the temporal cortex and in the hippocampus. During emotional processing, the functional connectivity between the voice area, the amygdala, and the hypothalamus was selectively enhanced in the context of blue illumination, which shows that responses to emotional stimulation in the hypothalamus and amygdala are influenced by both the decoding of vocal information in the voice area and the spectral quality of ambient light. These results demonstrate the acute influence of light and its spectral quality on emotional brain reprocessing and identify a unique network merging effective and ambient light information. Light therapy is the treatment of choice for seasonal affective disorder, SAD, and is a promising treatment for other major affective disorders, 1 and 2, suggesting that light can modulate mood in the long term. To better understand this effect, and because neural networks involved in emotional behavior have been implicated in mood disorders, 3, we first assessed whether light can acutely influence normal brain emotional processing. Indeed, ambient light is known to regulate processes other than vision, such as hormone secretion, body temperature, and sleep, but also alertness and cognition, 4 through 8. These non-classical, also called non-image forming or non-visual response, but see recent findings, 9. So these are references. The numbers, 1, 2, 3, 4, 5, 8, 9 are just referencing down at the bottom. Responses to light are mediated through a non-classical photoreception system, which is maximally sensitive to blue light, approximately 480 nanometers, as opposed to the classical photopic luminance visual pathways, maximally sensitive to green light, which is approximately 550 nanometers. And pathways maximally recruits the recently discovered intrinsically photosensitive retinal ganglion cells, which hereafter we referred to as IPRGC, expressing the photopigment melanomenism, in addition to rods and cones, which is 7, then 10 through 13. The impact of ambient light is detected in the longer term through the regulation of circadian rhythms, 4 through 7, and the benefit of light therapy on mood has been proposed to be mediated through a long-term circadian effect, which is 14. However, non-classical responses to ambient light also result in acute physiological changes. For example, ambient light significantly modulates ongoing cognitive brain function, including attention, working memory, updating, and sensory processing within a few tens of seconds, which is a big deal for the human brain. 6, and then 15 through 18. The amygdala, a core component of the emotional brain, 3 and 19, that receives sparse direct projections from IPRGC, which was the intrinsically photosensitive retinal ganglion cells. 20 is one of the brain areas acutely affected by changes in ambient light. 18. This result raises the intriguing possibility that ambient light directly influences emotional brain processing. Although this hypothesis may have major implications in basic and clinical neuroscience, it has not yet been tested experimentally. And the brain mechanisms underpinning the influence of light on emotional processing are unknown. Here we used a functional MRI, fMRI, and a validated non-visual auditory emotional test to characterize such a direct effect of ambient light exposure on brain responses to emotional stimuli. Results. 17 young and healthy participants, table S1, performed in the morning hours a gender discrimination task on verbal but meaningless emotional and neutral auditory stimuli. 21. Common but not completely identical pathways are activated regardless of the direction of the emotional valence. 22. However, our experience in the study of emotional responses is that negative valence elicits greater responses, which are less influenced by individual and variability in valence perception. 23. Therefore, half of the stimuli were pronounced with an angry negative prosody and the other half with a neutral prosody. It is known that these negative sounds elicit larger responses than neutral ones in the voice-sensitive area, 24, of the temporal cortex, and to a lesser extent in the amygdala, 25 and 26. Importantly, this effect is detected even when attention is directed toward the gender classification test and not the emotional content. The task thus allowed us to separate the known effect of ambient light on attention from its potential influence on emotion processing. While performing the auditory test, participants were exposed to 12 to 40 second periods of monochromatic blue, 473 nanometer, alternating with 12 40-second periods of green, 527 nanometer light of equal photon density. Figure 1. To identify modulation of brain activity by ambient light, brain responses recorded during the blue light exposure, to which the non-classical photoreception system is maximally sensitive, were compared with the activity recorded during green light exposure, to which the classical phototopic luminescence visual pathway is maximally sensitive. Behavior. Accuracy for the gender discrimination task was high and was not affected by emotional or light conditions. Performance was over 87%. F1 through 16 was less than 2.11. P was greater than 0.17. Figure 2A SI results. In accordance with the literature, 25, reaction times were slower for negative relative to neutral stimuli, indicating a significant effect of emotional valence. Individual ratings obtained after the fMRI sessions confirmed that perceived valence differed between emotional and neutral stimuli. Importantly, reaction times were not influenced by light conditions. As expected, given the short duration of light exposures and the low intensity used in the present experiment. 6. Critically, the effects of ambient light on ongoing brain activity can be detected with minimal exposures so that behavioral differences do not confound neural responses. Functional MRI. We first considered brain responses associated with a specific time point event corresponding to light onsets. Confirming our previous findings, 18. Responses in the right amygdala significantly differ between blue and green light onsets, which is in figure 3.3a and table 1. This differential response was primarily related to response adaptation across the session, i.e. across 12 light exposures, figure 3c. The average response to light onset averaged across the 12 exposures did not differ between light conditions, figure 3b. A composite representation of the average response and its evolution shows that the amygdala responses to blue light onsets were high in the beginning of the scanning session and monotonically declined throughout the session, indicative of a habituation process, whereas responses remained mostly unchanged for green light onsets. Figure 3D. We then considered the impact of the emotional valence of the stimuli on the brain activity. This also confirmed our findings, 25, that negative voices triggered stronger responses, compared with neutral ones. In the right inferior frontal cortex and bilateral superior temporal GYRI STG, within the voice sensitive area independently identified during a separate voice localizer fMRI session. Figure 1A, Table 1, and Figure S1, 25 and 26. So, significant impact of the emotional condition was detected in the amygdala, but this is in line with the observation that response to emotional response in the amygdala is weaker in the auditory than in the visual modality. 26. Critically, when considering the impact of ambient light condition on emotional stimuli processing, brain responses elicited by angry voices were enhanced under blue compared with green light exposure bilaterally in the voice-sensitive area of the temporal cortex and in the hippocampus, figure 4A and B and table 1. In contrast, no brain responses to emotional voices were significantly increased under green versus blue ambient light. Likewise, no significant difference between blue and green ambient light exposures was observed for neutral stimuli, and the sex of the participants did not significantly influence the results, SI results. We then conducted physiological interaction, psychophysiological interaction analysis to assess changes in functional connectivity dependent on ambient light condition. In short, PPIs, which is the psychological physiological interaction, tests for condition dependent modulations of functional connectivity by assessing the impact of experimental conditions, i.e. blue versus green light exposure, on the regression between the activity time course in a seed region and that of any other brain area. 27. We detected an increased functional connectivity between the voice-sensitive area of the left STG in both the left amygdala and the hypothalamic area, selectively for the processing of angry voices in the context of ambient blue light exposure, relative to ambient green light exposure, figure 4C and table 1. Similarly, the functional connectivity between the right STG and the same hypothalamic area was significant during the presentation of angry voices under ambient blue, but not under ambient green illumination. Finally, the protocol included two irradiance levels that were applied to both blue and green light exposures methods. Irradiance was not the primary focus of the present research, and only two low irradiance levels were investigated. Statistical analysis did not reveal any compelling effect of irradiance, table S2, and it seems unwise to derive from the present data a reliable characterization of the impact of irradiance on regional brain responses. Discussion. These original results demonstrate that ambient light and its spectral quality influences the brain processing of emotional stimuli, blue relative to green. Light increased responses to emotional stimuli in the voice area of the temporal cortex and in the hippocampus. Furthermore, in the context of blue illumination, emotional processing was associated with an enhanced functional coupling between the voice area, amygdala, and hypothalamus. These effects were detected in the absence of behavioral bias and were not significantly influenced by the sex of the participants. As in our previous research, 18, we reported an impact of the ambient light condition for the onsets of the light exposure, which occurred only once per minute and critically were independent of the ongoing task. We interpret this result as an initial high responsiveness to blue light onsets, which decrease with time owing to the habituation process in the emotional system. 22. Although the auditory task did not explicitly engage declarative memory, the hippocampus, which is also involved in fear conditioning, 19, was recruited by the auditory emotional task, and significantly more so in the context of ambient blue light exposure relative to green light exposure. Significant activity modulations induced by ambient light were already reported by the hippocampus at the onset of the blue light or after bright white light, while participants were engaged in attentional processes, 16 through 18. We surmise that ambient light indirectly influences the hippocampus through projections from the amygdala, 28, and brainstem nuclei, 29. As compared with green light, ambient blue light also amplified responses triggered by vocal emotional stimuli in the voice-sensitive area of the temporal cortex. To assess the potential mechanisms by which light can modulate responses in the voice-sensitive area. We tested whether its functional connectivity with the rest of the brain during emotional processing changed as a function of the light condition. Results revealed strengthened functional interactions under ambient blue light exposure between the voice-sensitive area, amygdala, and the hypothalamus, which constitute critical areas in emotional and light irradiance information processing. In other words, the responses of the amygdala and hypothalamus to an anger prosody stimulus were linearly dependent on the response in the voice-sensitive area in the context of blue relative to green ambient light exposure. These measures of functional connectivity suggest, therefore, that through their extensive recurrent connection to 28, the temporal cortex and the amygdala interact more strongly to process emotional stimuli in the context of blue rather than green ambient light exposure, thereby instantiating the interaction of light irradiance signal and effective signals. Functional connectivity changes were mainly apparent in the left hemisphere, and in line with the known difference in response dynamics in the left and right amygdala. 22. Adaptation is known to occur much faster in the right than in the left amygdala. 22. Potentially explaining why we did not detect sustained responses to vocal stimuli in the right amygdala in any ambient light condition. An anatomical connectivity could easily support rapid responses to light in the amygdala with a preferential reaction to blue light. First, the amygdala receives sparse direct inputs from IPRGCs. 20. Second, the amygdala also receives indirect retinal inputs through the supercolliculus and pulvinar 30, as well as via other brainstem nuclei 28. And we previously reported a greater sensitivity of blue light of the responses of the thalamus, including in the pulvinar, and brainstem related to auditory cognitive tasks 17 through 18. Only brain areas significantly involved in the processing of the vocal stimulation and affected by the light and emotional conditions could be detected in our event-related analysis. Therefore, the brainstem of the pulvinar could convey light signal without being detected in the present analysis. A number of hypothalamic nuclei are located in the surrounding of the detected hypothalamic area, including the ventromedial VMH and part of the dorosmedial DMH hypothalamus. The amygdala is in a position to control the expression of fear responses through its direct inputs to the paraventricular nucleus and the lateral hypothalamus. 19. Both receiving direct and indirect inputs, respectively, from the DMH and VMH. 31. The hypothalamus is known to receive few inputs from auditory temporal cortex, although auditory inputs can reach it by polysynaptic routes involving the amygdala. 19 through 32. The present study therefore strongly suggests that through its structural and functional connectivity, the hypothalamus is a potential site of convergence for emotional 19 and 33 and ambient light information 4 and 6. The wavelengths used in the present protocol were chosen according to the spectral sensitivity of the classical phototopic luminous visual pathways and of the non-classical melanospinnic-based photoreception system in an attempt to separate their respective influence. However, several mechanisms can be contemplated to explain the spectral influence of ambient light on emotional brain processing. First, the fact that light of shorter wavelength triggered significant modulations of brain activity is an argument in favor of the involvement of the non-classical photoreception system. In addition, we are reporting light modulation of brain responses elicited by auditory stimuli, modulati, which are in essence non-visual. To our knowledge, there is no evidence that visual responses to light modulate brain responses associated with a gender classification of auditory vocal events in the context of constant diffuse light exposures. Finally, we report sustained responses that span several tens of seconds, whereas visual response are typically transient and time-locked to changes in visual signal at the level of photoreceptors 1034 or neural ensembles of the optical cortex 35. For these reasons, we favor the implication of the non-classical photoreception system in our effect. However, this interpretation would entail a significant influence over radiance. The present data do not allow us to reliably characterize the impact of irradiance on regional brain responses, and our hypothesis shall await new experimental data to be firmly confirmed. Second, visual mechanisms could be responsible for the reported effects. For instance, the prevailing preference to blue hues in the general population could have contributed to the differential responses between blue and green light exposures. 36. However, performance on detail-oriented visual tasks, i.e. tasks that, as in our experiment, require focused and careful attention, is enhanced by red relative to blue hues. 37. In contrast, blue compared with red hues seems to increase creativity and innovation. 37. On the basis of these results, which did not include green, one would therefore expect a larger impact for colors associated with longer wavelengths, i.e. green, in our experiment, whereas the contrary was observed in our experiment. Third, color opency recruits retinal and cerebral mechanisms, 38, that seem to participate in the nighttime suppression of melatonin secretion by light, 39, or in pupillary constriction, 40, when exposed to polychromatic light. Similar effects might have taken place in the present experiment, although the alteration of both monochromatic light exposures and the 20 seconds of darkness between the exposures potentially reduce the influence of color opency in our results. It is even more challenging to assign the reported effects to the recruitment of specific photoreceptors. All retinal photoreceptors are probably involved in the effect we describe. 11, 13, 41, and 42. And we have no means to isolate the contribution of any of them. Melanospin expressing IPRGC play a key role in non-classical responses to ambient light exposure. Their maximal sensitivity, 460 to 480 nanometers, is close to the peak wavelength of blue light, 473 nanometers, used in the present experiment, and the light levels we used are compatible with the threshold identified in rodents for their recruitment, 43. Our results are therefore compatible with the contribution of melanospin expressing IPRGC, and rodents' rods also mediate non-classical effects of light at radiance levels higher than previously expected, 43 and 44. Their contribution cannot be ruled out, although their maximal sensitivity, 503 nanometers, is intermediate between the blue and green, 527 nanometers lights used in the present experiment, in reducing their potential influence. We cannot exclude a potential contribution of short wavelength cones, S cones, maximally sensitive to approximately 420 nanometer wavelengths, which have been integrated in previous models of the non-classical impact of ambient light, 45, and shown to send input to some IPRGCs, 34. Finally, M and L cones, maximally sensitive to approximately 530 nanometers and 560 nanometers respectively, as well as S cones, could have contributed through the putative color opency mechanism already mentioned. Novel experimental designs are required to evaluate the respective impact of the visual and non-classical photoreception system and of the different retinal photoreceptors in the modulation of the emotional brain responses. As a whole, our results support the view that ambient blue light promotes effective arousal and associated mnemonic processing, which may favor a rapid turnover of limbic reactivity to emotional challenges, and thus could participate in a rapid behavioral adaptation to the environment. 6. Emotional responses are acute, transient phenomena triggered by external stimulations, whereas mood is a sustained emotional state. Changes or alterations in moods, as much as in mood disorders, modify emotional brain responses, whereas responses to emotional stimuli can have a great impact on subsequent mood. 3. Importantly, mood disorders such as major depressive disorders and bipolar disorders are characterized by altered structural and functional changes in areas involved in emotional processing. such as the amygdala, hypothalamus, and hippocampus. 3. And we observed an impact of ambient light exposure on responses related to auditory, emotional stimulation of these areas. By strengthening emotional brain reactivity in these areas, ambient blue light in the responses of the thalamus, including in the pulvinar and in the brainstem related to auditory cognitive tasks, might promote accurate and contrasting responses to emotional signals, which could ultimately enhance efficient, good mood regulation processes. In fact, recent reports showed that prolonged darkness or lack of light in rodents reduces a depression-like state associated with structural brain changes, 46, whereas the complete blindness increases depression risk, 47. Likewise, long-term changes in ambient light daily profiles, such as light therapy, seem to restore normal mood regulation, 1-2. And the spectral compensation of light changes across the seasons, 48. Interestingly, blue-enriched light seems to be equally effective as visually brighter white light in the treatment of SAD, and a polymorphism in the melanospin gene has recently been associated with SAD. 50. Collectively, the data show that the spectral composition of ambient light influences the processing of emotional stimuli with a superiority of blue light in recruiting a network merging effective and ambient light information. For auditory voice stimuli, this circuit involves the amygdala, the voice-sensitive area, and the hypothalamus. Although acute effects of ambient light on emotional processing might differ from its long-lasting effects on mood, the present findings in healthy subjects may have important implications for our understanding of the mechanisms by which changes in lighting environment improve mood, not only in mood disorders using light therapy 1 and 2, but also in the general population using blue-enriched light in the work environment. 51. Although traditionally these effects of light were thought to be related to changes in circadian rhythm parameters, such as the timing of the melatonin rhythm, our data suggests that they could also depend on brain mechanisms that can swiftly modify brain emotional processing, potentially through melanospin-based non-classical photoreception. And the methods cited, and then the acknowledgments, and then the reference page. So let's talk a little about what they're saying. It is saying that, yeah, blue light contributes to productivity, but it also contributes to emotional response, which it falls in line with what we know about businesses. You know, I switched from incandescent to fluorescent lighting, and partly because fluorescent lighting offered more in the blue spectrum, which made the workers more productive, but also made them irritable and gave some people migraines. So there is, you're overstimulating those parts of the brain, and so there are going to be negative results as well. So it does wake you up and make you more productive, but it also makes you extra moody, because you basically feel all these different, especially negative. They focused heavily on negative responses in their tests, but yeah, like you sense it more. You basically feel it more so. So it's a mood enhancer as well. That's interesting. That's very interesting. But it can also be treated to reduce depression. Is what they're saying as well. Light therapy. So it's got lots of uses. It's also got some warnings. Like you have to find a middle spot. You know, where the good and bad. The good hopefully outweighs the negative. That's what the report is saying. And this report, this research paper is led by the same guy as the last one, Giles. It's is earlier though. Like the one that we read before was much more recent. This paper was from 2010, so at least 9 years ago now. But it shows that he's been writing about blue light for well over a decade, because some of his earlier papers we'll get into some of them, and they're from the early 2000s. So look forward to that. I think he's got a lot to share. And if you'd rather I start at the early papers and work my way up, just let me know. Or if you want to just keep going back this way, we can do that as well. All right.